In this last video for this lecture, we're going to talk about just a few of the most important features of antibodies. Antibodies are also referred to as immunoglobulins. They are produced by a descendant of the B cell, which is called a plasma cell. Antibodies are considered humoral molecules. They are not cells by themselves. They are proteins that are produced by plasma cells. Remember, the plasma cells come about after a B cell gets activated by an active T cell. Now, these specialized proteins, these antibodies, literally have the shape of the letter Y. This shape produces what we refer to as the two arms of an antibody and the stem of an antibody. Now, the arms are referred to as the FAB, and I'll explain that terminology in just a minute. And the stem is referred to as the FC. So when you look at an antibody with its Y shape, there's going to be a place on the FAB, there's going to be a location on the arms where the antigen binds. That's where FAB comes from, antigen binding. That means that the structure of the arms has to vary from antibody to antibody to antibody because each type of antibody that's produced from a plasma cell is specific for that particular antigen that triggered its production. In other words, the place on the antibody that binds to the antigen will bind only to that antigen. The stem is very different. The stem is the constant region of an antibody. That's where the FC comes from. C for constant. All antibodies inside your body, all of the antibodies that get produced by your body, have the same type of stem structure. And that's to allow those antibodies to stick to the surface of certain immune cells, just as we saw in the last video when we could see antibodies stuck to the surface of a memory cell. So, antibodies will be produced by plasma cells and will circulate in the body. They will circulate in the blood and in the lymphatic systems, and they will also enter tissue. Antibodies are specific. They are capable of reacting to that particular antigen that triggered their production. Now, there's some terminology around antibodies that's important to know in clinical medicine, and that's these terms monoclonal and polyclonal. When you hear an antibody described as monoclonal, what that means is that it's an antibody that was produced by a single plasma cell. In other words, all of the antibodies that get produced by a single plasma cell are going to be identical to each other. Their FAB region is going to bind to one particular epitope on one particular antigen. Remember, we said an epitope is just a region of an antigen. Monoclonal antibodies are antibodies that all come from the same plasma cell, a single plasma cell, and all have the same FAB. Polyclonal antibodies are different. Polyclonal antibodies are produced by multiple plasma cells, and they're going to all have different FAB in that they can bind to different epitopes of the same antigen. So polyclonal antibodies, they're all still going to respond to the same antigen. It's just that they're going to stick to different epitopes of that antigen, different regions of that antigen. So you can have a monoclonal response, you can have a polyclonal response. Let's take a look at the structure of an antibody. This is just a diagram, but this is quite literally what antibodies look like. They have a Y shape. They have what we call a stem and two arms. Now, you'll notice that there's some colorization to this drawing. There's this sort of turquoise color here that's labeled as a heavy chain, and there's this sort of fuchsia color here that's labeled as a light chain. Don't worry about that terminology. That's not important for us to know. What is important is that you understand that the FAB is located here and also here on the arms. And the FAB is that place where the antigen is going to bind to this antibody. The FAB is specific for one particular epitope of one particular antigen. The FC is different. The FC is the region right here on the stem of the antibody that is able to bind to immune cells, the surface of immune cells. So in other words, for some immune cells, like memory cells, this part of the antibody will bind to the surface of the immune cell so that the arms are reaching out off the surface of that cell. And if that cell encounters the antigen will bind to the FAB. Now, there are different classes of antibodies, and I've got them listed here. You may have heard of these different classes. The IG designation in these abbreviations stands for immune globin or immunoglobulin. Remember, that's just another word for antibody. So there are immunoglobulin M type, there are immunoglobulin G or IgG type, there are IgA type, IgD, and IgE. Different classes of antibodies are found in different parts of the body. Different classes of antibody are involved in different types of immune responses. We haven't talked about rapid immunity versus delayed immunity versus allergic immunity. That's not as important for us to understand for a class like this. But just understand that there are classes of antibodies that have slightly different responsibilities in terms of how they function in the human body. Antibodies are proteins. And remember, proteins have a limited lifespan. They will exist in the body for a certain amount of time, and then they'll be degraded. All molecules in living cells have a limited lifespan. But when it comes to the immune system, that means something important. That means that antibodies have to be replenished over time. Antibodies are not going to last forever. Now, you may have been tuned in to the news about the pandemic virus that we're all living with right now, the SARS-CoV-2 virus, and the race to develop a vaccine against this virus. And you may have heard the news that there's some concern about the human immune reaction to this particular coronavirus. There has been some reports that people who've been infected with this virus develop antibodies to it that don't last very long. In fact, the most recent research suggests that the antibodies that are produced against the SARS-CoV-2 coronavirus may only last for about 3 to 6 months. That's a problem because that at least suggests that people can get infected with this virus more than once in a single year. That's quite unusual. That's something we don't see in other viruses. So, as we work to develop a vaccine against this virus, the vaccine, which will help produce antibodies against this virus, is going to have to be able to elicit antibody production that's effective to help keep us safe from this virus. And right now, we don't know whether or not the vaccines that are in development are going to produce antibodies that only last for 3 to 6 months, or maybe antibodies that last a little longer. We just don't know yet because all of this is happening for the very first time. But what you should understand as a student of microbiology is that anytime antibodies are produced, whether by a natural infection or through vaccination, those antibodies are only going to last for a certain amount of time. And when antibodies don't last very long, that means sometimes we can be reinfected, and sometimes we need to be vaccinated more than once. Now, as we listen to news stories about the vaccines that are currently in production against this pandemic virus, I want you to listen for a particular word regarding antibodies, and that is the term neutralizing. The best that we can hope for with vaccines is that they stimulate the production of what is called neutralizing antibody. And that's because, like the name suggests, neutralizing antibody is the kind of antibody that best helps eliminate pathogens like the SARS-CoV-2 coronavirus. Let's finish up with this summary of the adaptive immune response. T lymphocytes, or T cells, are going to be presented with antigen from invading pathogenic microbes. The cells doing that presentation are macrophages and dendritic cells. The T lymphocytes are going to bind to the antigen that's being presented to them. That binding is going to cause the T cells to differentiate, to start maturing and dividing into different types of T cells. It's also going to stimulate the T cells to produce and release various humoral chemicals. And the combination of those things is going to activate B lymphocytes, or B cells. The B cells will then mature or differentiate and divide to produce plasma cells and memory cells. The plasma cells are going to produce antibodies that are specific for that particular antigen, for that particular pathogen. The memory cells are going to keep the memory of that antigen. Those memory cells will then be able to respond immediately the next time that pathogen tries to infect the body, the next time that antigen is detected by the memory cells. The memory cells will rapidly stimulate the plasma cells, and they will produce again those specific antibodies very quickly. Finally, the final action of the adaptive immune response is the destruction of the pathogen. The antibodies that are produced will bind to the antigen on those pathogenic microbes, and it essentially marks those cells or those viral particles, just like a flag. The pathogenic microbes that are marked with antibodies, marked with those flags, will then be immediately recognized and destroyed by other immune cells.